We are Group E1B1 and I am Nathan. Here is the summary and approach of our project. We changed the aim to extracting DNA from various fruits due to certain limitations. Our group did research on the topic focusing on how to conduct an experiment. A practice experiment was conducted. We then researched on the importance of the steps in the experiment. We then repeated the experiment.
I'm Sanjeev, and this is some background information for our project. Deoxyribonucleic acid, or DNA, is the blueprint of all living things. DNA is mostly found in the nucleus of cells, and it is a type of nucleic acid. DNA is a polymer. The monomer units of DNA are nucleotides, making DNA a polynucleotide. Each nucleotide is composed of a 5-carbon sugar, also known as the deoxyribose, a nitrogen-containing base attached to the 5-carbon sugar and the phosphate group. There are 4 types of nitrogenous base, namely adenine, guanine, cytosine, and thymine. DNA exists as a pair of long molecules intertwined with one another, giving rise to its double helix structure.
I'm Clifford, and here are the apparatus and materials needed for this experiment. You will need the Ziploc bag, statistics, test tubes, filter funnel, sieve, measuring cylinder, glass rod, test tube rack, dropper, lysis buffer, and ice-cold ethanol. The materials needed are the fruits we are going to be using, namely strawberry, lemon, apple, rambutan, kiwi, and blueberry. There are variables in this experiment. The change variable in this experiment is the type of fruits used. The fixed variable, which are variables that are kept constant, are the mass of fruits, volume of lysis buffer added, volume of ice-cold ethanol added, and the concentration of the lysis buffer. The measured variable for this experiment is the amount of DNA extracted from the different types of fruits.
I am Keegan. This is the procedure of our experiment. Step 1. Check the mass of the 6 different fruits using the electronic balance, ensuring that we get 20 grams for each fruit. Step 2. Place the fruits in Ziploc bags separately. Ensure the Ziploc bags are tightly sealed and proceed to mash the fruits thoroughly. Step 3. Mix 20 ml of lysis buffer into the different Ziploc bags containing the mashed fruits using the measuring cylinder. Step 4. Mix the lysis buffer with the mashed fruits in the Ziploc bags by pressing the contents. Step 5. Filter the different mixtures from different fruits and collect the filtrates in separate containers. Step 6. Use a dropper and add 9 ml of ice-cold ethanol into each container drop by drop, releasing the ice-cold ethanol from the dropper gently at an angle. Step 7. DNA will precipitate out. Use the blunt ends of satay sticks to twirl and spoon out the DNA.
I'm Jessima, index number 1, and here is the result and analysis of the experiment. The ranking of the fruits are as follows, from the most amount of DNA extracted to the least. First comes the blueberry, followed by kiwi, strawberry, lemon, apple, and rambutan. From our experiment, we found out that berries have more extractable DNA compared to other fruits because berries are generally soft and easy to destroy. Also, berries have lots of genomes. For example, strawberries are octoploids, which means they have 8 copies of each chromosomes. That's why some fruits have more extractable DNA than others.
I'm Jayi, and to conclude everything, we rely on iPower to compare the amount of DNA extracted from different fruits as the school lab does not have a spectrophotometer. We have also found out that berries contain a lot of DNA compared to other fruits.